Genomics and gene editing technology. Genomics is an offshoot of molecular biology concerned with the genome's structure, function, evolution, and mapping. New technologies to manipulate genes in cells are in development. Companies like Pfizer used these technologies to create vaccines for COVID-19 quickly. This technology also treats cancer by reprogramming cancer cells. The potential for improving the quality of life is virtually limitless. But how far will this technology grow, and what concerns could arise? In 1997, Sony Pictures released a movie called Gattaca. In this movie, people edit their children's genes to make perfect people. These genetically perfect people view naturally born humans as second-class citizens. This movie was science fiction at the time. Today, the ability to edit genes is becoming science fact. In his article on gene editing, Robert Sparrow discussed the ethical implications of genetically modifying humans. Not only would unmodified humans be seen as lower class, but the latest generation of modified humans would also see each previous generation as lesser. Similar to how computers become outdated as newer technology becomes available, successive generations will have to contend with becoming outdated and obsolete. Viewing this scenario in the Kantian principles of ethics, one could say that we should always strive to improve ourselves as a species. With that thought in mind, editing our genetic code and improving the technology with which to change our genetic code is seen as a moral imperative. On the other hand, treating others as lower class citizens based on their genetics is racist and morally reprehensible. One could view the difference between genetically modified and naturally born people as a split between two separate societies. A moral pluralist way of thinking lets the two societies coexist while allowing such moral injustice to continue. Treating naturally born citizens as lesser would be the norm in the group of genetically modified citizens. Of course, the natural born citizens would have differing views about the high-class population. Another ethical issue seen in genome editing is the non-identity problem. If you have the technology to detect and remove the genetic code responsible for congenital disabilities, shouldn't you use it? In their article, Alonso and Savulescu discussed He Jiankhui's experiment with twin babies in 2018. Having the ability to edit a person's DNA before they're born, one would assume that they have a moral imperative to use this technology to prevent the person from being born with a disability. Editing the DNA before the person is born raises the question, is this the same person that would have been born without the genetic editing? Setting aside ideas of predestination or other concepts impossible to chart, the person has different DNA from the potential person that they once were. The moral implications of removing one person from existence and replacing them with another with nearly identical genetics are mind-boggling. Scientists could use genome editing to clone human organs or even full clones. Charles Vicanti seemingly grew a human ear on a mouse's back. The public thought this was unethical genetic tampering. Vicanti achieved the effect by injecting cartilage cells into a mold on the mouse's back. Genetic engineering could grow an ear, heart, or liver. Organ recipients could receive a perfect match. The science of gene editing is quickly becoming emerging technology instead of science fiction as genomic technology improves. Where is the organ coming from if we create just an organ for transplant? Are we producing it in a lab? Is a living organ donor paid for growing the organ like a surrogate mother? Dr. Sakar answers some of these questions in his article on cloning. Instead of requiring a whole donor, organ tissue could be repaired or grown for the organ recipient. Cloning the tissue within the organ recipient makes the patient both the donor and the recipient. A small sample of the organ tissue would be required to clone the organ. Stem cells are added to the tissue sample to grow into the corresponding organ with cells that are a genetic match to the original, with zero chance of rejection. The stem cells take the form of the original organ's cells and form a patch to repair or replace the organ. Another approach to creating a genetic match for an organ transplant recipient is to clone the recipient. DNA from the nucleus of a cell can be transplanted into an embryo with nuclear DNA extracted to produce an entire cloned organism. Some couples commit to having an additional child to serve as an organ donor for an older child with a terminal illness. Geneticists test the zygote's DNA to ensure they will be a genetic match and will not have the same fatal disease as their older sibling. Having a child to prevent the death of an older child, let alone cloning a child to save the older one, goes against Kantian ethics. Cloning a complete human being also raises several problems. Are clones a new human or the same as the original? Are they twins or copies? If a clone kills the original, is it murder or suicide? In the absence of data, we must draw our conclusions from what information is available. Currently, couples can spend money on in vitro fertilization. We don't call these babies petri dish babies. Using this logic, we can conclude that clones are new humans on par with twins. Having established this status, if a clone kills the original person, it is murder. If a person asks their clone to kill them, one could say they used their clone for assisted suicide. Some may consider clones less than humans, but we should afford them the same rights as we have. In conclusion, the science of genomics and the technology used to edit genes are not new concepts. The science and technology of 1997's Gattaca are quickly approaching out of science fiction and into science fact. With the prospect of genetically improved humans comes the possibility of societal split. There'll be schools of thought pushing for genetic modification and others against it. With the advent of gene editing technology, cloning will be a possibility. We should treat clones like we treat other humans, with respect. Thank you and have a good night.